So I will talk about like using microfluidics for optimizing gamete and embryo handling. So why do we need that? And why? So there are microfluidics devices developed. So I think the first microfluidics for assisted reproductive technologies has been described in the early 2000s. But until now, we don't use that in the clinic. So there were devices that were developed for cryopreserving oocytes, for doing in vitro maturation, devices for sperm separation or selection by like gradient of, for example, progesterone for in vitro fertilization and in vitro culture, and also, of course, some more like fancy devices for like not just embryo culture, but also doing on-chip analysis. But even though we have all these devices and been over like 25 years working on that, at least in the reproductive field, it's still not well used in the clinic or in the field. And then I think like there are some reasons why for that. One is complexity. So for fabricating the devices until a few years ago, we need like fancy equipment, clean rooms that can be really costly and technically demanding. Integrating into the clinic required changes to normal protocols, training, regulatory clearance, which makes it harder. So you don't want to risk something that has been working to a certain extent to change for something that can bring risks. So there is a lot of steps until we can integrate those into the clinic. Of course, scalability and standardization. So it needs to be consistent, reliable performance is needed for the routine use. And so far, we don't have anything in that there. But I think for me, the most important thing that what I'm going to talk now, they're difficult to introduce or remove gametes in there. In those devices, you need practice for that. And also there are some challenges to the use, like leaking, bubbles formation, so not being suitable for field applications. So you need to the pump or whatever it is in there. So when I talk about field applications, that is something that really interests me, especially now that I'm working with the zoo. So when we go to do collections of gametes in the field for like, doesn't matter which kind of a species, it's really hard. So here is an example of what we bring. So we bring all the equipment we need. And well, sometimes in the field like this, what you see here in this picture is a jaguar in there that was collected. It's a really good condition because you can go with the car, but sometimes you need to hike 8-10 kilometers under rain, whatever it is. And carrying equipment is not easy. So for the past few years, we have been working on optimizing field equipment. For example, we have now portable, battery operable warming plates, as you can see here, or a mini fridge, or like a tube holding that keeps the temperature. We have like a sperm analyzer that is a little lens is connected to an iPad. But we still lack a lot, especially like when we collect semen, we need to wash away the seminal plasma and place them with the extended and the cryoprotectant solution. And it's not possible, like, because then you have to bring a centrifuge and some other equipment to the field, which makes it difficult. So that's why came the idea of using producing devices in which we could wash and select the sperm in a field setting. And that is what I'm going to start first. And then the next, I will talk about the oocyte side. So first, we'll talk about the device for sperm and wash and selection that was done by these two students from my lab. One was a visiting student from Brazil, and Roxanne is a PhD student in the lab. Unfortunately, I cannot disclose the device. We are still validating some steps for the patent. But it's basically a filtration device with different compartments where we add the semen, and in a couple minutes, literally like a minute or two, we can wash that semen with any solution you want and then collect it with whatever solution you want. So to test that device, to produce that device, we use a 3D printing. We produced molds with the 3D printer. We treated those molds. I think Bastien can talk a lot about that. He helped us a lot with that. We cast the PGMS, cure it, and then bond them with plasma. The reason why we chose to do 3D printing is because I gotta tell you, we have tried so many different devices. So if we were going to do the normal, the more convenient way, it would be so expensive and not possible. And while with 3D printing, we can change the design in the same day, have a mold ready and a device ready to be tested. So that's why we chose to do 3D printing. So next, we tested this sperm washer with bovine semen. So we collected the epididymal sperm from bovine from the slaughterhouse, and then we tested different concentrations of sperm. And then we compared the device wash with normal centrifugation. That is what people do. And then we check for concentration recovery, the motility, so how the sperm are moving, and some other parameters. Sorry. So here I want to show you in purple, we have centrifugal devices. In yellow, the devices used in the wash, in our washer. So what is interesting here is that with lower concentrations, we have a recovery that is lower than normal centrifuge, but when we go to the higher concentration, we have similar recovery. That indicates to us that we need to make devices of different sizes for depending on the amount of concentration. It's something we've been working on. So for now, I will focus only on the 300 million per ml concentration that is the one that is compatible, because we are still validating the smaller devices for different concentrations. So with that, we also check the total motility. So we have the initial one, so it's the fresh sperm collected, and there was no difference in the motility, and also comparing to the centrifugation. We check it for progressive motility. So progressive is when the sperm swim like in a straight line. That is really important because things like that sperm is the one that has the most chance of fertilizing the oocytes. And there was also no difference comparing to the fresh and the centrifugation one. Again, with lower concentration, it was a problem, but we are working on that. We then check it for viability, just so it was the nigrosin staining. So when the sperm is pink, it's because the membrane is damaged, so it's not viable anymore. When it's not, when it's like transparent, it's viable. And again, we'll focus on the 300 million. There was no difference in viability between the fresh and the centrifugation. The integrity of the acrosome. So we did a staining for the acrosome, that is this cap on the sperm head that is really important for fertilization. And as you can see here, is a normal intact acrosome. So here are 3 sperm heads, and one of them had a normal acrosome, the other 2 didn't. And then we saw there was no difference on integrity of acrosome for the 300 million. Of course, for the other ones was a little bit different. And then we also checked it because when we were analyzing those semen, we saw a lot of decapitated heads. So oh, sorry. Oh. So as you can see here, there is a head without a tail. So we check it for that because we have a feeling that it was more, but at the end it was the same amount. It was more than the initial, so the fresh one, but was the same with centrifugation. So we were pretty happy with this device because it was fast and easy to use. So instead of having to centrifuge your semen for like 5 minutes, 10 minutes, depends on the species, you can in a minute wash it, resuspend that with the media you want. You don't need centrifugation. We do need to change the size according to sperm concentration. It's something we've been working on now to see, like, have different size and see the range of concentrations that are good. We have similar recovery to centrifugation and also similar quality as centrifugation, which is really nice because then that gives us an opportunity to use that in the field or in the clinic without having to centrifuge the sperm. Next, I will talk about the sperm selection. So when we were thinking about sperm selection, we were thinking about different ways we could do that, and we have tried. So that's why I say using 3D printing is nice because we have tried so many different designs, and a lot of them didn't work. So, but then one really worked well is what I'm going to talk to you now. Unfortunately, again, I cannot disclose the design, but I can tell you we were working on the sperm thermoattractivity. So the sperm normally in the female tribe would swim towards a warmer environment. So we tried to make a device where we could create this gradient of temperature. And then because we wanted something to be field deployable, I think, and with my veterinary background, what we found was like using nichrome wire embedded in the PGMS and then controlling that with a voltage controller, we were able to warm up that device. So again, we prepared then with 3D printed modules and preparing then. One thing that we had to do, and I can disclosure, the PGMS is not really good at dissipating the temperature homogeneously. So we were checking, and then one thing that came to mind was to introduce aluminum hydroxide in the PGMS, so it would help spread the temperature. And that's what we did. So we started with different percentages of aluminum in there that also helped us create this gradient that we were looking for. Again, so just we introduced the sperm with a syringe, then we collect the sperm from there, and then this was the controller that we were using because it's easy. We can just connect that to a power bank. And I cannot show those images now, but we check a temperature. We have a nice gradient of around half degrees Celsius. And then, so we were fine, happy with the model. So we decided to test that with the sperm. So to test the sperm, I again use a epididymal sperm from bulls. We use a 50 millions per ml in this case. And then we compared that in two different groups. One group was inside the device. And then we have the device inside the incubator. So the device will be there and we will not have this gradient of temperature. We checked that the temperature is consistent. And we have the device on the bench. And then we compare that with the swim up. The swim up is like you centrifuge the sperm, you make a little pellet and you place media on top. And the sperm that is good, they will swim to the media. And because we were using our devices, this hip stop that is a normal sperm handling media, we did the swim up also with this media, but also with a commercially available media, just to be sure we are comparing with the best we can. We again check it for recovery of sperm, motility and progressive motility, and some sperm characteristics. So here, first I want to show the recovery. So we have the device in the incubator, the first purple. In blue, we have the device controlling temperature on the bench. In green, we have the embryo cloud, the commercial available sperm selection, and then the swim up with the same media used in the device. What was interesting in the recovery is that when we have the device with this temperature gradient, we have a higher recovery of sperm compared to all the other groups. When we check it for motility, it was also higher. In this case, we have row here is the seeming pre-selection. So the motility was higher, was higher than the row, but not significant, but was higher than the two swim up methods. And then progressive motility was also higher in this case, and even higher than the raw semen, which is what we want. We want to select the best sperm. We then checked again viability. There was no difference in viability between the device controlling temperature and the raw and the other methods. We checked for the integrity of the acrosome. And then what we saw was there was a reduction in acrosome integrity. So maybe we might be capacitating this sperm. It's something we're still figuring out, but we think this gradient of temperature for this 1 hour that we do is capacitating the sperm. But what we were really, really interested and happy to see is that the number of fragmented sperm reduced to almost zero, of course not, but was much reduced compared to all the other methods, showing that we are selecting good sperm with that device. So just as summary, this device is also easy to use. Well, it's not fast, it's 1 hour selection like we normally do for the swim up. There is no need for incubators, so we can just have that controller in there with like a power bank. We have a higher recovery than swim up, in better sperm quality, especially in progressive motility and lower DNA fragmentation. We also check the oxidative stress, but because I have a short time today, I didn't show that data, but we also reduced oxidative stress in there. The other one negative aspect we saw was reduced intact acrosome. So maybe it's capacitating that sperm is something we are investigating. We are going to investigate further. So, okay, we have these two devices that seem to be easy to use and field deployable. So we decided to test it. So we have collaborators in Brazil that does a really nice work in jaguars conservation. So we won in Brazil this past March, testing those devices. So we have jaguars that were collected by pharmacological sperm collection. I will show you what that is soon. And then we did get the sperm at 100 million per ml for the washer and then checked all the characteristics. And we also tested the selection of the jaguar sperm because the jaguar, we didn't have much sperm for that. And we also wanted to preserve those. We didn't compare with swim up or centrifugation in this case. First, because we didn't have the equipment where we were collecting those samples. And second, because we didn't want to waste the samples. But we did check two media for the sperm handling. PBS is what they normally use with the jaguar sperm just to wash it after collection. And beyond, there is extender for bovine and equine that we were testing if that would work for the jaguars. And then again, we check it for those characteristics we talked before. We also did some sperm where we wash it with the washer and then select it. If you're curious, this is the Jaguar, this is Conservation Institute. So that was not in the field, it was our first trial. We didn't want to do that in the field, but was in a place where we didn't have a lab. And so this is how we collect the sperm from the jaguar. We insert a catheter in the urethra, and then we do some stimulation of the glands, the prostate and other glands in there, and then the sperm, the semen just coming. If you can see here, white is semen, and every time you do a stimulation, you can see the semen coming out. So we did that, and then we check it for the recovery. So I will start with the wash it. So washing the jaguar sperm was really variable. So we have some recovery that was really low, but some recovery that was almost 100%. I think it depends a lot also on the concentration of the semen. But we also have a really good recovery for selected. And in pink, we have the Beyond, just commercial media, in blue, the PBS. So we were washing with PBS, that is what they normally do. And when we did selection, we selected with the Beyond and PBS, and we saw the Beyond, this media that is used for like extending the semen and then refrigerating it, was much better for the Jaguar. So we have a really good recovery of the semen, even better than the bull. Some males was like over 60%, which is a lot comparing other available models for selecting sperm that are similar. They normally select between 15 and 25%. So we were really happy with that. And then we also had a good selection when we wash it in and then select them. We next check it for the motility, and then we compare to the fresh collected semen. That is here the first one. And there was no difference in motility in the selected ones, but then with the Beyond media. But then there was a reduction in motility when we washed them, and then also when we selected with the PBS. So we know PBS for the Jaguar semen long term is not good. It's fine for washing, but it's not good for keeping the sperm. We then checked for progressive motility, and what was really interesting is when we select the sperm, we have a much higher motility, progressive motility, even than the fresh. And in fact, there was one animal, that is this one here, that had almost 99.5% motility, which was pretty good. So what we saw was that the washing process damaged the sperm a little bit, but when we selected that, we recovered it. Of course, we're losing concentration. We then checked for viability, so using nigrosin staining, and there was no difference with the fresh one, so the process was not damaging the sperm. Just the washing process was reducing the viability, so we need to optimize that. We checked for acrosome by the pulp staining. There was no difference in acrosome integrity, and we also checked mitochondrial potential in this semen. And we also didn't see any difference compared to the fresh. We are still finalizing, analyzing the data for the DNA fragmentation and also morphology of the semen. But we were really happy. That was our first try with a different species that we haven't tried before. And it seems to be working really fine, especially the selection. The washing, we are damaging some, but we still not bad. Like we can recover that if we select it. So that gives us a little bit of a tool to use that next time we are going to Brazil to collect with huge animals. So next I want to talk to you about the oocyte. So collection, wash, and maturation. So that was the work done by Roxanne, a PhD student in the lab. So we basically was recently published on LabOnAChip. So we basically made this device with microwells in there, to which what we were trying to do here was to be able to get the oocytes inside the device without having to search for them in the stereo microscope and be able to wash and everything without needing any equipment, then a syringe and a pipette to get the follicular fluid in there. So what we do in this device, we add the follicular fluid here, and then by like pulling and waiting for them to settle, we are able to trap the oocytes in there. That's all we call that, oocyte trap, and then change the follicular fluid to culture media, and sorry, and then in vitro mature the oocytes in there. The same as before, we made this device with 3D printed mold. We also have maybe 10 different designs before we got to this design. And this is how it looks like. So we have a reservoir here. This is a 5 ml tube. It is a bigger device, but the volume inside the microwell area is the same volume we use in a Petri dish, in a 4-well dish for oocyte maturation. And then, so we have a dam kind of structure. So it's going down to direct the oocytes to the microwells. They're looking like that. And then there is like a wall here. So here is higher. So the cells in the oocytes should not be trapped out. So we did in vitro maturation in the ootrap. So as I said, we collected the follicular fluid from oviries from bovine from the slaughterhouse. We added them in the reservoir. I will show you a video how we do that. And then they were trapped. And then we did in vitro maturation inside the oocyte that under perfusion, 20 microliters per hour or static. And we have a 2D control, just a normal 4-well plate. Then we check it for integrity of the metaphase plate and spindle. That is an indication of the quality of the oocyte and that is matured. And then we also did RNA sequence. So here's just a quick video, so how the device looks like. So here in yellow is the follicular fluid. We basically just grab that in and do that. We do move that sideways a little bit to make them to not just get stuck in the middle and help also remove some of the debris from it. And so from both sides, and then after all the follicular fluid is in, we can add the wash media and then wash away. And then we can change to maturation media and then get that inside the incubator for culture. And here is how it looks like from the inside when we do that. So as you can see here is follicular fluid getting in. It's a lot of cell debris. And we don't remove all those cell debris, as you're going to see later. So when we get the culture media in there, and this is how it looks like. So we do have a lot of cell debris and a lot of microwells. They don't have oocytes. They just have cell debris from the follicular that comes from the aspiration of the follicle. So here with the yellow arrowheads, we can see the oocytes. And just quickly, here is a quickly time lapse of the 24 hours maturation. You can see here the oocyte, it had some other cells in there. So the cumulus cells from the oocyte is just this, but those are other follicular cells. And so you can see they expand and they kind of give this compression in the oocyte. But so far, we don't think it's a problem to have these extra cells first when they're inside the follicle. They are growing those extra cells. They're growing with different forces in there. And so we didn't care about having those extra cells. Also because when we saw our results, we were confident that we were not damaging the oocytes by having them there. So when we have perfusion, we have a higher rate of matured oocytes than just the 2D control, the normal Petri dish. Was even higher when we have the static oocytes in there. So not just, I think, the stimulation from the perfusion that was really low, but also the compression and then being a different stiffness matrix were also helping them mature. What was really interesting to see is that normally when you mature the oocytes, we have like a metaphase plate where you have the chromosomes aligned and then a bipolar spindle here marked in green, and then the extrusion of the polar body. But sometimes that can be abnormal. Like here we have the chromosomes aligned in 3 different places. There is a misaligned chromosome and it's not a bipolar spindle, it's like a 6-polar spindle. So what we saw when we have the oocytes culture there, under perfusion, we have a lower, although it's just a tendency, we have a lower rate of abnormal spindle. So like having this multiple, not just bipolar spindle, and also chromosome alignment, which was really nice because those are really important for further embryo development. We were happy with that, so we also wanted to do like a field deployable device. So what we did was to make a smaller version of it, in which we also incorporated nichrome wires, and we also 3D printed a syringe pump that was connected to an Arduino step motor and Arduino controller, that we could do everything without needing like just a power bank, but without needing a bigger pump or an incubator. So this is how it looked like. We checked for the temperature inside the wells, inside the microchannel, and then we saw that around 40 minutes it stabilized the temperature. And then here is our 3D printed pump. So with that, a small can be contained, and we just added HEPAs to the maturation media that we know can support maturation. So here is the one controlling the voltage for the temperature, the pump with its Arduino controlling that. And we were able to, in that device, to have, again, higher rates of mature oocytes compared to the normal 2D, and also lower number of abnormal spindle and chromosome alignment. So we were really happy because potentially, for example, for farmers where they go and do OPU, like oven pickup in the field, normally they have to have a lab nearby to do the in vitro fertilization. But with a device like this, we give them 24 hours to transport those oocytes to a lab to do in vitro fertilization. And the same would be for field collection in the females that are possible to do, such as like bisons or different wildlife that we can do that in the field. But it's not just about fields. We were trying to understand what was happening with those oocytes. So we know that during the follicle maturation, the follicle growth and oocyte maturation, the follicle, there are a lot of forces happening there, like hydraulic pressure, compressive stress, tensile stress, hydrostatic pressure. So it's building up fluids inside the follicles that is growing. And also after, when it's ovulated, fertilization happens, there are a lot of forces in the reproductive tract going on, such as shear stress by the fluid flow, and also the characteristics of the fluid, such as viscosity, can change these mechanical forces, compression by muscle wall in the oviduct, for example, in the endometrium, friction of these embryos with the cells, vibrations from the beating cilia. So there are a lot of forces in the female tract that when we do in vitro fertilization and embryo production, we don't mimic them. So to understand that better, we first analyzed mechanotransduction. There are like pathways that sense those mechanical forces and translate into response between in vivo and in vitro produced embryos for cow in different stages. And what we saw was that most of those pathways that are like mediated by piezo receptors, integrins, TRPs, or GPCRs, they were upregulated in vivo and downregulated in vitro in most of the different stages, with exception of some pathways that were actually downregulated in vivo. But interesting, since you're talking about oocyte maturation, 94% of these mechanotransduction pathways, they were upregulated in vivo, meaning that yes, during maturation and yes, during embryo development, these mechanical forces are modulating this embryo. What are the responses? We don't know. It's something that was not really well investigated, but it's something we've been working on. But we then thought, okay, in our device, we are kind of mimicking. So this tensile and compressive stress, we can mimic, as you saw, those cells growing and then pressing the oocyte. And there we have shear forces from the flow. We have hydraulic and hydrostatic pressure. So was that changing our oocytes? So we compared, we did a RNA sequence from those oocytes, and we compared 2D, dynamic, and static. So when we compared 2D versus static, there were just two genes that were different between them. But when we compared the 2D and the static with the dynamic, there were over 250 genes that were upregulated in there. So because we were interested if we were mimicking those mechanotransduction in these oocytes, we also focused on analysis of those mechanotransduction pathways. And what we saw was that between 2D and static, there was just few significant pathways. But between dynamic and 2D and dynamic static, there was around 40% of those mechanotransduction pathways that were upregulated in the dynamic compared to the static situations. And then just here is a summary. So we have a higher piezo and piezo pathways, integrating responses as well for different pathways. So we really have to see, even though we were not bringing that to the level of the in vivo, we were rescuing a little bit those mechanotransduction that are missing in normal in vitro situation. So now we've been doing embryos in those devices, and then we are just waiting for the RNA sequence. The embryo rates are the same, I gotta tell you. We didn't see improvement in embryo development, but we wanna see if we can improve embryo quality. Just a summary. So OTRAP is fast and easy to use. So normally you would collect oocytes, you would search that under stereo microscope and wash, and that would be a process that can take something between 40 minutes to a couple hours. In here, we can get them inside the device in few minutes, like 3 to 4 minutes. There is no need for a stereo microscope, not incubator, if you use the heated version. What is really interesting, if you want to do some dynamic toxicological exposure or hormonal exposure or whatever exposure, we could do that because we have a dynamic system, or we can just do everything in the incubator without like removing them out of the incubator and transferring them. We can do everything inside. What I really like it is that we are inducing these mechanical forces in there, and we have higher oocyte quality in maturation rate, lower spindle abnormalities, and we do have an in vivo like mechanotransduction. It's not to the same level, but it's similar, it's better than normal in vitro. And with that, I would like to thank you for your attention.